everyone. Welcome. Thank you so much for being here today with us. I am Dr. Mickey McComb-Kobza. I'm the Executive Director of Ocean First Institute. We are a nonprofit organization in Boulder, Colorado, and our mission is ocean conservation through research and education. Today, I am joined by Dr. Misty Paig-Tran. She is a biomechanics professor at Cal State Fullerton, and she is here to join us today to talk a little bit about bio-inspired designs. She runs a lab and does research on manta rays. She's worked on oarfish. She has worked on Amazonian fishes, sharks, rays, you name it. Misty has worked on it. So it is an honor and a privilege to have you here, my friend, today to talk about your amazing research and your work. So we're going to do about 20 minutes of a presentation. And as you're listening in the audience today, if you have questions, we'll be taking questions after the presentation. You can type your questions into the Q&A box. And at the end, I'll go ahead and take your questions and ask Dr. Misty the question. So we're going to go ahead and get started. I think she's going to go ahead and share her screen. And again, thank you so much for being with us today. And she's going to go ahead and get started.
Okay. And I think we have this. Yeah, cool. I think the screen, hopefully everybody can see it already. And thank you for having me here. I'm going to call you Dr. Mickey for today, but or Dr. McComb. It's really exciting to be invited by you because, of course, you're my hero and I love you. So I'm very excited about that. I follow your work closely. All right, so bioinspiration from the sea, and hopefully I did only have one cup of coffee, so I wouldn't be too excited because I get really super excited about this and sometimes go a little fast. So I'm going to try to go nice and slow here.
Okay, perfect. I'm not sure I can see your screen.
Oh, no, is it not?
Maybe you can try one more time.
I can try it once more and do the share.
My end. Just wanted to let you know that I am able to see it on my end.
Oh, great. Okay, perfect. Then we're good.
All right. So bioinspiration from the sea. Just to get started with that, really, my lab is, as Dr. McComb was saying, more of a biomechanics lab, although it just sort of fortuitously, we have gone through our research with biomechanics, and we often actually find bioinspiration from the sea without actually looking for it. So that's kind of the theme of this talk is if you sort of look at the world as a naturalist and you ask lots of questions, that sometimes those questions can then translate into practical uses for human use. So if I click on this, there we go. Just to start off with what is exactly bioinspiration, and probably this is very familiar to anyone who's watching here, but just to give you some ideas, this is the idea of looking to nature to then inspire new designs for human use. And so probably some people are familiar with things like soft robots, at least in marine biology. This is the new hot trend. So using things like hydrostatic skeletons, that means pieces of animals that don't actually use bones to move, so bone muscle connections. Instead, they use water connections. So the best hydrostatic skeleton I can ever think of is always an elephant's trunk. And so we can use that sort of technology and that performance of an elephant's trunk to then create new bio-inspired robots, specifically robots that don't get crushed under deep sea pressures. Another great one is the idea of using avian bone, so bird bone, to help us to build new buildings and structures that are very lightweight, but also very strong. So probably the one bio-inspired design that everybody is familiar with is the idea of gecko adhesion. So looking at a gecko toe, which you can see over here on the left, and all of those little hairs then help to create this really strong adhesive forces that allow people to go up things like, glass on buildings. And the idea here is that if we can build bigger and better suction cups, then people like window washers or others that have to go very high will have a better connection to the buildings and create a much safer climbing environment. For me, I always think that's great. I love human health, but I also think a lot about myself because I'm very selfish. And so I think, well, why can't we use this to build something like a new shower caddy that's not going to actually fail in the shower? And so hopefully we're very close to that. And I'll show you in the next slide why I say that we're very close. But if you think about it, you know, almost every suction cup on the market currently will fail. And that's because it gets an air pocket or water that then disconnects that connection. So my former advisor actually is Dr. Adam Summers, who's up at the University of Washington. And he's actually the person who sort of got me into Bioinspiration just on a whim. So one of the things he studies is clingfishes. And so you can see here's the disc of the clingfish. Hopefully you can see my arrow pointing to it. And even in dead clingfishes, so here's a dead clingfish and here's a dead clingfish, they still suck really well. And they not only suck on smooth surfaces somewhat, but they actually suck better on rough surfaces and wet surfaces. And so just by looking to nature and understanding that clingfishes can suck really hard on rocks in the intertidal, he was able to then translate that into new technology. And I'll just show this quick video here because really, I love it. That's a dead fish, and it's picking up an apple that's over 80 times its weight. So really super strong technologies, and I love his laugh at the end. So he and his postdoc Petra then have created new suction cups, and that's kind of the theme of this Bioinspiration. They weren't out looking for new technologies. They were simply out observing nature and said, I don't understand how that works. So that's exactly what my lab is doing as well. Just to give the audience some idea of what the scope is of my research. So you can see here are the first three that I'm basically going to talk about to some degree in this talk. So my work with manta rays and filter feeding and how that has then translated into new technologies. I have a grad student who finished recently who was working on fish bone, and then also another grad student, both grad students named Andrew, who worked on tiny armors and catfishes in the Amazon. But my research really spans a really wide swath of different topics, from sand crabs digging to antibiotics from the sea, how to basically pick yourself up on your teeth if you're a walrus, to whale hearing, and microplastics. So we're really kind of interested in just all things marine and how we can translate these marine animals and performance into these new technologies potentially. So here is my grad student down below. I'll point to him with my pointer. That's Raj Divi and myself, and Raj should be finishing up actually today, hopefully, with his master's degree. And so this is some work that we've done on manta ray feeding. Now, my PhD was basically completely on manta rays, and Raj picked up where I left off and really figured out how a manta ray is able to filter feed on the smallest animals in the ocean, even though it's one of the largest animals in the ocean. So just to play a quick video, and this isn't mine, I pulled it from Blue World. They're much better at photography, like Dr. McComb is. I'm terrible at it in the water. I get too distracted. But what you can see is these mantas are filtering using what's called ram feeding. That means that they open their mouth and they swim with their mouth open, and they're basically continuously filtering plankton. And so it was with that, just watching mantas feed, actually via video, that I started asking questions about manta rays in a class when I was just graduated from my undergrad. And some of the questions I asked were, well, okay, how does such a big animal actually feed on these teeny tiny little plankton? It just didn't make sense to me how they were able to get enough of this plankton to help their body grow and change. And so you can think of whales and other very large fishes, all are plankton feeders. And if you looked to the literature, which I did, basically everybody thought that they used this sieve filtration, which means that just like a pasta and a water colander, as you pour the pasta through a colander or a sieve, the water exits and the pasta is going to remain. And that's basically what every filter feeder in the ocean what we thought that they did. Only recently was there another mechanism called crossflow filtration that has been described that may also come into play. So in my head, as a new grad student or almost grad student, I thought, okay, so they filter and they're using a sieve, and that kind of makes sense, except for I didn't really see these manta rays closing their mouth and sort of coughing or clearing any clogs. And one of the things about sieve filters is all sieve filters will clog eventually. So there had to be some clearing mechanism. And occasionally you'll see a manta kind of cough, except for in water, but not with enough regularity that it would make sense that they were using that type of filtration. So just to give you an idea of what that looks like, here's a manta ray, here's the inside of a manta ray's mouth, and here's the plankton. So the filters are actually right here in this black, and this is the gill arch. That's what holds the filter. So you imagine this, this, and this are all filters. When I actually looked and started doing some anatomical measurements on these things, I noticed that the filters were actually, the pore of the filters were much bigger than the plankton. So if we go back to that analogy of the colander, that would mean the pasta would just go right through the holes. So something wasn't making sense. I did try to go out into the wild and put basically a camera inside of a manta ray's mouth. So you can see that this looks very soupy. It's very sort of greenish blue color on here. And that's just laden with zooplankton. So that's me trying to film a manta ray. And I'll show you what this basically looks like. Me coming with a camera, the manta sees me and then hauls the other way. And this happened over and over and over. Whale sharks are happy to let you put a camera in their mouths. Manta rays are just not going to do it. They have one of the biggest brain to body ratios. They're incredibly smart, as Dr. McCone has told you lots of times, I'm sure. They don't like to be bothered. And so I had to come up with something new to actually figure out where those particles were going and how they were filtering. So what can you do if you can't get live video? You go to models. And that's what I did. So I started to look at the morphology. So over here on the right, you can see this is actually scanning electron microscopy. What that means is I had the filters from museums of manta rays and other mobulids, which are their close relatives. And I would look at them under really high-powered microscopy to see what the surface of the filters look like. And so you can see over here, some of them had these tooth-like structures called denticles across them. Some of them are very, very smooth. And some of them, this is a manta ray down here, have these little finger-like projections that project into that, that pore. That would be like a colander pore. I could then use the measurements from the anatomy to create models of what these filters look like. So this would be one of these lobes here. So that's a lobe, and that's the hole or the pore. And we could do it in actual size, and then we could actually blow it up to 4 times the size, because if you blow it up, you could actually slow down the water and really see what's happening. So we moved to these models in order to figure out what was going on, and we filmed them in a flow tank. So here is some video. This video was actually taken by my undergrad, Ashley Peterson, who's now a PhD student over in UCI. But what you can see, if I start this video, is that sieving is definitely not happening. If sieve filtration was happening, you would expect that the water would actually be coming in from the top of the screen and down. But the orientation of the filters aren't conducive to sieving at all. Instead, what I mentioned earlier, that crossflow filtration looked like it was potentially the mechanism for filtration. And so what that means is particles are going to come in with this flow, and instead of diving down into the pores, they're going to be concentrated at the back. What I wasn't expecting are these little, and I'm going to play it one more time. Oops. These little things down here. So these little eddies completely, I should say that again, I'm so excited. They took me completely by surprise. I wasn't expecting them. I didn't know what to do with them. And I started thinking about what they could be doing. And I thought, okay, we have these cyclones. I think this is something completely new. I've never seen it in another filter feeder before. And so I started thinking it must work kind of like your Dyson vacuum cleaner. where particles would come in and start spinning around, and just through centrifugal forces, right? Just like if you're on a ride at the fair and it's a spinning ride, you get thrown back to the side. Like that's the same kind of principle as what I thought was happening. And I actually went on NPR and I talked about that, and I said, you know, I think that this is what's happening. And the worst thing you can possibly do when you have a really good idea is to keep testing. So I unfortunately am that person who decided to keep testing rather than keep my opinions, you know, just to what they were. And so I said, okay, well now I need to put some particles in here and just confirm that this is the case. And so here's some video of what I saw. There's some particles you can see bouncing, 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 and they never get caught in that cyclone. And that took me a long time to really hash out why that was. I kept throwing particles in there. Or I should say I kept making my poor grad student throw particles in there over and over and over. I think he did this close to 300 different times. Always with the same result. The particles would bounce off of those filters and not ever get entrained in that cyclone. So I knew that this cyclonic filtration couldn't be what was happening. Instead, we had to, and I will go there, start to model this. And so I'll show you what's happening. Those are the filters, and water and food particles will move in. And as water starts to exit, those particles actually get really close to that little recirculating cyclone, and the cyclone actually speeds up the particle. So it speeds it up and it slams it into the next filter. And when it slams it into the filter, it actually then ricochets off of that filter and then back into that flow up above, which goes back towards the esophagus. And this happens over and over and over so that you get that bouncing effect. And so we thought, cool, that really, you know, solves some things. It answers the question of how can the Manta filter out particles that are much smaller than the pore size. So it sort of takes away that sieve idea. But there are no other filters on the market that work like this. And so we had this challenge of, number one, we had to name it. And we really had to figure out what the mechanism here was that was driving this. So we had it in video, but we needed to do some more mathematical work. And so for that, I brought in James Strother, who I used to have a picture on there, and I took it off, and I wish I still had it there. But Dr. Strother, who is now over at University of Florida, he did some of this work, this computational work, and what he found was, so if you look over here on the right-hand side, the red are the particles and they're ricocheting. We were able to recreate that computationally, so with a computer. We were able to recreate that recirculating flow, and the water leaving in blue. So we could get back to the mechanism real easy. And what we found is we could get almost 100% filtration efficiency with particles that are much, much smaller than the pore size. So here we have a pore that's 1100 microns, and we're filtering at almost 100% at about 400 microns. So incredibly small comparison. And there just really are no other filters that are like that on the market or otherwise. And that was really exciting for us. So we've now named this ricochet filtration because of the ricochet effect as those particles elastically recoil off of each of those filters. We know that we can get much, much smaller than the pore size. We knew we had something new, so what did we do? We decided we were going to patent this, right? So, and we're currently still in the end stages of our patent. It takes a couple years to do that, but we now have a patented design based off of manta rays. So you can see up in the upper left is our early design. We've now since adjusted that. This is actually to show you up here, we're doing it all over again. So now we want to know what's happening with whale sharks, because whale sharks are said to use crossflow filtration, and I don't buy that. And not because it's not a great idea, I think it's awesome, but because now we've done some calculations and we find that we are getting very similar numbers with our early calculations as manta rays. So I'm wondering if they're not doing something similar or something completely different. Whale sharks are huge. So we've we've now found this completely non-clogging, high efficiency, large volume filter. There is absolutely no other filter on the market that does that. All filters suffer from some sort of clogging effect and cost energy to unclog those filters. So, so I'll put, our next steps is we're starting to work with wastewater treatment and other types of filters. So for example, the artisanal beer companies have a lot of microplastic pollution in them, and we're starting to work with those different types of entities in order to place these filters in and see if they are a good solution to clogging filters. So we have about 2 more minutes before I'm set to answer some questions. So I just wanted to quickly give you some other highlights of what is going on in my lab. So for example, I have armor, bone, and skin all happening. Armors based on fishes is really not a new topic. We've been doing it. That's an armor from the Han Dynasty there, and I just, anytime I can put Jason Momoa in a talk, I'm gonna do that. So we have some fish armor here, but most fish armors are actually based on really large armored fishes like the Arapaima here. My grad student happened to notice that there are these little tiny catfishes down here. So there's the teeny tiny catfish that's about the size of my tip of my finger to my first crease in my finger. It's teeny tiny, that we had some reports could survive piranha bites and be totally fine. And so in the lab, we actually tested that. So here is just some video of piranha, because why wouldn't I show you piranhas biting something? And this is actually a juvenile, so this is not an adult piranha, and it already delivers 15 pounds per square inch of pressure, which is enough to basically cut through my hand down to the bone. I have all my fingers. I'm totally fine. Never got bit. But that's a lot of pressure to be put on a teeny tiny little fish. And so what we did, and I'm actually going to skip this video because I don't want to freak anybody out, some young audiences, but what we did was we actually tested it with a fish, and that piranha would bite and bite and bite on that fish and never penetrated the armor. So they have this really crazy flexible armor and really strong armor. So just to put that in perspective, it's kind of like having medieval armor where it's really strong but not very flexible, coupled with like a Japanese samurai armor that's super flexible but not really strong. And this thing is doing both. It's kind of crazy. And the way it's able to do both, just to give you some of the highlights, is it actually has this nice synergism between the really strong the scale and the soft tissue underneath. So the two play together, and if you remove one of those, it fails completely. So we can puncture through it really easily, but together they form a super highly strong armor. And so the idea is, for me, I actually started thinking about how can we translate this into human use, and my idea was gardening gloves. For me, it was actually fishing gloves, but gardening gloves work too because I do both. I was tired of getting hit by spines on rockfishes, but also when I go out and garden, you know, if you're using gloves for, let's say, trimming your rose bushes, they're really thick and bulky, and you can't really get a good tactile feel on what you're doing. The trade-off is you have thinner gloves, but you feel all the pokes and pricks through it. So the idea is, how can we then translate this to new gardening gloves that are really flexible, but also really strong? So that's what we're currently working towards at the moment. And then just to give you the last 2 highlights, I had another grad student who was looking at fish bone, and most fish bone is actually very different than, let's say, human bone or other mammal bones. Most of them are what's called acellular, which means that they don't have little cells in their bone. And so 60% of the world's vertebrates are fishes. The majority of them are acellular, but we have about 8 papers now that actually say anything about acellular bone. And so my grad student had questions about whether or not that bone frees well, like mammalian bone, and if it doesn't, could it be used potentially for new grafting for humans? So if you have a bone graft, if you get that bone tissue from another mammal, so like a pig bone, which is common, or from another human, the problem is there's foreign DNA. And so if you have a bone that doesn't have cells in it, you're more likely to have less foreign DNA. So that might be a good solution for grafting into humans without having humans have to be on immunosuppressants for the rest of their life. And so he actually found that the bone freezes incredibly well. It holds up mechanically, so it's a really strong potential for bone grafting. And then the final thing I'll leave you with what's coming out of my lab, and really out of my collaborators' lab as well, is we're working on antimicrobial, or I should say antibiotics from the sea. So there's this always this war between antibiotic resistance and finding new antibiotics. And the sea is an incredibly diverse place with lots of different biomes within it, but it really isn't tapped as a potential for antibiotics, not very well anyways. And so we've started to look at the skins of fishes, especially in really sort of mucky areas like the LA harbor. And surprisingly, these fishes basically all have antibiotics on their skins. We have found, and she might not be happy with me saying this, some really good potentials for new antibiotics. And in fact, with one of our fishes, we have a metabolite in there that's produced in one of the bacteria in their skin that inhibits carcinoma in colon cancer, right, in humans. So I think that that's really, I'll sort of get punny and say the new wave of antibiotics research that's going to happen. I hope that more people start to look to the ocean because it's a really, really huge potential for new antibiotics. And so with that, I will kind of leave it at, here are my students, and let you ask questions since I'm at my time.
Oh, that was amazing. Wow, thank you so much. We have tons of questions here. People want to hear some more from you specifically. So again, thank you so much. That was amazing.
Oh, you're welcome. Thank you for having me.
Yeah.
I'm so excited for the questions.
Okay, so here we go. Mika wants to know what are the patented filters going to be used for? What kind of applications do you see this in? And seems endless, but what do you see?
Yeah, so Mika, that's a great question. For me, the reason I was convinced of doing this was for wastewater treatment. So one of the things my lab works on are microplastics in the ocean and also in things like oysters and other food fishes. And what we're finding in our early results is that the microplastics that are really out in at least our coastal environment are a lot of fibers. And these fibers are actually coming from washing your clothes. So there's a lot of synthetic fibers in clothes, and they are not treated in wastewater treatment plants. So after most everything else is treated, they are then going out into our coastal environment and are exactly the same size as plankton. So fishes are actually uptaking them, and it's a real problem. So we are working with Orange County Sanitation District to some degree already to potentially use our filters in their systems. My husband is the other person on the patent besides Dr. Strother and I, and the reason I brought him in is because he's a big business person. And so he was like, I drink microbrews, I like, tequila and things. And one of the things when I went to a microplastics conference was I have a colleague who was testing all of the microbrews from microplastic contamination from their tubes. And unfortunately, just about every microbrewer there is still has microplastics. So you're ingesting them if you're of age. Probably not so much for sodas right now because those are the bigger players, but the smaller players have a real problem with microplastic pollution. And so he really wants me to target those applications, and I'm sure I'm totally happy to do that.
That's amazing. There's a lot of microbrews in Colorado, and certainly one of the big benefits is having local people involved in making these microbrews. So certainly having microplastics in the microbrews is not a good thing. So
no, it's a great, and they always taste so much better. I hate to say that, but they're so good. So yeah, anything we could do to help them out, and it would be, in theory, a simple fix for them. So
great.
We're hoping it works.
That's really, that's fascinating. So I have another question here from Anatole. I hope I have your name right. Saying very interesting, and this is regarding your Manta research. In the end, where do the particles go? And talking about the ricochet. What about the remaining water and the inflow? What percent of clean water could you get if you use that for filtration? So kind of a two-parter question there.
Okay. For the mantas, those particles actually end up back at the esophagus. And so what happens is, hopefully you can kind of remember, since I'm not screen sharing at the moment, that the water is going to exit out the pores and the particles then are going to bounce to the back. So there's actually very little water that remains back there. And what happens is then about 10 minutes in or so, the manta will then close its mouth, squeeze out the last bit of water and swallow the particles. And so that's where the particles go, back into the tummy. And then the second part was about
how clean
the filtration, the clean water. So our hope is, because we've done some of this a little bit, once we're above a certain range at the moment, we're about 100% effective. In theory, is that always going to be the case? No, we've got to manipulate a little bit right now. And we're actually playing right now with getting smaller and smaller particles. And we can do that. We just have to ramp up the speed of the water. So at some point it becomes limited by having a pump that will pump water fast enough to do that. But yeah, at the moment, if you're over about 400 microns, the particles, and you have an 1100 micron system, you're 100% efficient, and then it goes down from there. So computationally, we can get down to 30 microns, so the size of about bacteria, and get to that efficiency as well. We just have not actually done it with my models yet because of COVID. So that's what we're supposed to be doing right this moment.
Wow, incredible. Another question that's kind of related, and this is, it's kind of funny. It's just like, why did you even decide to make the sieve based on manta rays? Like, what was that you saw that made you so fascinated with manta rays?
It actually wasn't really manta rays that I was interested in. The way I started this was I took a course right after I graduated at Friday Harbor Labs, which is where I met my PhD advisor. And he was going into a lecture all about fluid mechanics. And it was something, even though I had a marine biology degree, I hadn't really thought about fluid flow through fishes. And he was talking about the difference between like large scale flows and flow through pipes. And they're very different things because pipes have wall effects. And so basically water clings to the side of pipes and changes the flow. And so I asked a really simple question, and I almost didn't ask it because I was pretty sure he was going to think I was like the dumbest person in the room, which tends to be the case. I'll come back to that in a moment. But, and I just said, what about in something like a whale shark that has a huge pipe? Would you get pipe effects in that? Or would it be, more like it's a big flow? And he stopped and was like, I don't know. And so luckily it wasn't too dumb of a question, but he was like, I don't know. Do you want to come and study that? And I was like, yeah, okay, sure. I'll do that. And that's actually how I got into my PhD, which is a terrible way to decide to go and do a PhD, but it worked out. And so I actually not only worked with the mantas, but I worked on whale sharks and also on basking sharks and megamouth shark. I just kind of picked the manta rays to focus on for this talk, even though I've worked on the other ones, mostly because I know the most about manta rays.
That's amazing. And so tell us about the megamouth shark. What did you discover and what's going on with that? Do they have bioluminescence? What is the deal?
Oh my God. Yeah, megamouths. Yeah, so that's how I treat megamouths, is usually a sigh. They're really weird. I can tell you I have not made much progress. And the reason is their filter structures actually are completely covered by denticles, but not the same kind of denticles as mantas. They have denticles that look more similar to the outside of shark skin. And so I cannot wrap my head around how they're actually filtering, because they basically work kind of like a big baleen whale where they'll take a big scoop of water and then process it, but they don't have mucus and they are completely covered by denticles and the water sits in there for a long time. So that's about as far as I've gotten, is the morphology, and modeling it. I'm like, every time I pick it up, I just kind of throw it back into its container and decide that that's another time, because it's so weird. And yeah, they have bioluminescence on them. And why do they need bioluminescence if you have plankton? Well, plankton do come up to lights, so that kind of makes sense, but like, they're so weird.
That's amazing. Oh my God, we could talk
They're
forever
so
just about that one thing.
Oh yeah, and their bodies are like, so I work with oarfishes and other mesopelagic fishes too, which their bodies are basically like jelly.
Yes.
They really don't have mineralization in their bone. And megamouths are the same way, even though they're cartilaginous. I mean, you get one and it's dead, and the body just sort of slumps over, so you can't get good CT scans because it's basically like it deflated.
Like a big balloon.
So yeah, they're I mean, in theory, at some point I'm going to get to it, or somebody else is going to get to it. But yeah, they really have me baffled, and that's really not a hard thing to do. I'm baffled all the time. So
well, that's what science and nature is, isn't it? It's trying to understand how things work, and you know, you've got an amazing career asking these questions, and then, you know, trying hard to make a difference, you know, in helping with plastic pollution and microfibers. I mean, I can tell you that we have students here in Colorado that go out into the rivers, and we're sampling for microfibers and microplastics, and we're finding it. And
of course,
you know, it's really sad, but you know, how amazing that you're using nature's inspired designs to really make a difference. I mean, that's really nature saving nature. I think that's really cool.
Yeah, and it's always better at it than what I could come up with without looking.
It's so true. It's so true. It is just incredible, the mimics and things we can learn. Here's another question for you from Brittany. She says, I know there's a lot of work happening with aquaponics and mangrove trees to solve pricey salination issues, but as you said, clogging is an issue. Is it possible to use a filter like this to remove salt?
Salt would be difficult. And so the reason I say that is because you're really looking at it from, in theory, if it's in the water, right, then it's going to be dissolved in the water, which makes it just basically the element, you know, just small molecules. And so where we're at now is about 30 microns, which like a 1 NaCl, right, molecule that would be in there would be much, much smaller, because we're talking about it at the molecular level. So we have not gotten to desal yet. In theory, we probably will not get down to that level, just because we would be limited to by the pumps. So as we sort of need to filter out smaller and smaller things like viruses, then you actually have to speed up the water to levels that would probably be unimaginable. So like 500, you know, miles per hour kind of issue with, and so pumps are not gonna, like, once the pumps start getting their act together and creating pumps that work better, then that would be a possibility. But right now, we're really sort of limited on the technology that are available to push the water as well.
Nice.
It's a great question.
Yeah, that's a great question. I have another one from a friend of ours, Julius. Thank you for tuning in. He's an amazing artist. Oh my God, I'm so
one thing I can't do.
Yes, he's amazing. So his question is this: given their interesting hydrodynamic properties, have you or your students worked on various kinds of shark denticles for flow modulation? And I have a follow-up question after that.
For flow modulation, no. And the reason why we're not doing that is because George Lauder at Harvard is very into this. He actually posts basically like daily to weekly videos on exactly that, of all different types of sharks. I mean, he's done makos and everything. And so he's like kind of taken on this challenge with gusto, and he's got much fancier equipment than I am. So I'm kind of sitting back and watching it. So we haven't done too much with it, although we have done it a little bit with our filters. So adding on denticles onto our filters and seeing what happens. And it turns out that it actually is probably creating more flow separation earlier. So it stalls the flow earlier and changes the sizes of the particles that are being used depending on the type and size of denticles. So we have done some of that. I didn't present any of that. And my grad student is actually in the process of sort of finishing up that work so that he can write that up.
That's great. Well, just a follow-up question on that, and this is something that I think is kind of been well documented, is the use of shark skin and denticles in surgical tubing to kind of help resist, microbial growth, things like that. Is that something that is widespread in use? Is that something that was just kind of a novel one-off? Do you have any updates on that?
I haven't seen really any of the applications that have come out of that. It's certainly an interesting idea. And I know, I don't want to say I know, but I'm pretty sure that Marianne Porter has looked into that a little bit as well.
Okay.
Her lab has with the anti-fouling. And Marianne, I hope I have that right, that you were the one doing that. I know we've talked to you about it. I just don't remember if you actually were the one that did it.
Nice.
I'm pretty sure. But I haven't seen any like tubing on the market or anything that has really made use of that technology, at least not widespread. And I'm kind of running through the technologies that I've seen with the denticles. I mean, certainly Speedo tried to use that a little bit for their swimsuits, and that really didn't catch on either. I think it was actually banned from the last Olympics. They weren't allowed to use the shark skin suits, and so maybe that's why it didn't catch on, because it was banned. But for tubing, yeah, like it makes all kinds of sense, and I think it's just the roughness scale and the flow through the tubes would have to be right for that technology. So it wouldn't work with very low flow, but it could be implemented.
Interesting. Well, I have one more question, because I think we're kind of getting close to time. So thank you so much. But I'm really fascinated by your work with the catfish and piranha. And I am just a sucker for any South American fish. I lose my mind. But it's so fascinating to think about the arms race of, scales and denticles and armor and all of that. And I love this idea of the garden gloves, because I myself get ripped up when I'm in the garden. Where are you at with that? What are your thoughts about using more fish as kind of your bio-inspiration for something like that?
Yeah, we're not far along. we know the mechanism. We're still actually doing some work with them, because it turns out that when the fish is alive, it works really well. But unlike Adam Summers' dead clingfish that still cling, when they're dead, it totally breaks down. And so there is something going on between their muscle contractions and a sliding mechanism that's happening that we have not been able to recreate in the lab yet. And that's what we're sort of, the next steps of that are trying to really recreate this, because it really is dependent on a very finely tuned soft versus hard ratio. Mm-hmm. And even if it's dead, it still has a soft to hard, but it doesn't work anymore. And so getting that just so is key to getting those, any application for gloves to work. As soon as we figure that out, and if we can do it with an easy fix, then it's just putting them on the gloves and making it happen. So that is a much easier step. I would say don't look for awesome gloves inspired by the catfishes in the next 3 years. That's probably, I'm not that fast, especially not with the current state of things. But hopefully we're going to keep chipping away at it. If we can get back in the lab over summer, we're certainly going to make that one of our priorities, because I'm just really fascinated by how this teeny tiny fish can withstand piranha bites. It's just insane to me.
Yeah, that is absolutely insane. And then the very last thing, I just totally blew my mind talking about the acellular bones in fish and the possibility of using that in grafting. That's, mm-hmm. I mean, that could be a huge game changer for so many different reasons. How exciting. And
yeah.
I did not really think about that. So that's a new one for me.
Really interesting, right? Because we've thought for a while that cartilage would be similar, because cartilage also doesn't have, cells. Well, it does have it. I don't want to say it doesn't have cells. It does have cells, but it has fewer cells. But the problem with cartilage is it does not withstand freezing events. And so you fundamentally have to be able to freeze a tissue in order for a grafting event to happen, because you don't want to be harvesting it right away necessarily. Although some do. So there are actually 3 ways to harvest tissue for grafting. One is to harvest it from yourself, and so that happens as a surgery, but that actually kind of creates more problems than you would expect, because now you've got 2 injuries instead of 1. And then you can harvest from another human, which oftentimes that happens from somebody when they pass. Not always, but yeah, then you're on the immunosuppressants, or you harvest from like a pig or something that's a very similar, and again, immunosuppressants. So this is looking really promising, not only for the grafting, but also for studying just bones and fishes, really. I mean, it seems like maybe that's not as exciting when I'm giving a bioinspiration talk for human use, but for my purposes, knowing how fish performance is a big thing. And so doing mechanical studies on bone has always been sort of limited to fishes that are really close by, because you, in theory, can't freeze it because it would change the mechanical properties. And so my grad student showed that fundamentally that is not true with acellular bone. You can freeze it, you can freeze thaw it. He actually did some tests on bone that we got from grocery stores and live fish, or not live fish, but freshly caught fish. And he saw no difference in the mechanical properties. So now all of a sudden it's like, I could go out and test fish from Indonesia and figure out what their performance is. And so like that to me is even more exciting.
Yeah.
I hate to say it because I'm a marine biologist, but but like so much exciting, because it just is totally fine. So we're excited.
Oh, that's fantastic. Well, yeah, that does certainly open up a whole new universe for you for your research capabilities. So that's fantastic. Well, I think we have come to our end here, and I just want to take a minute to let you have any last thoughts or anything that you'd like to share. But I just appreciate your time so much. I know you're very, very busy, so thank you for making the time today to be with us and to share your story with all of our viewers.
It's my pleasure. I love talking science. Hopefully you got that from this. I get really excited. But the one thing I would say is just, everybody is a scientist. I know that doesn't sound right. Sometimes we think of scientists as having a PhD, but really we're all scientists. We're all naturalists to some degree. And so I always tell my students, and in fact, I told students yesterday as well, for lack of a better way to say this, I'll just be blunt about it. I feel basically stupid every single day. There's something every day that I don't know. And I hope that it's clear from this talk that that's totally okay, and you shouldn't know everything. And even if you think it's going to be a dumb question, it probably isn't. And so you'll either get your answer by Googling, or figure out that nobody else has asked that question. And, you go down 15 years of work on manta ray filtration, right? So, so I guess the point is just like keep asking those questions, keep looking at things and saying, I don't know what's going on, right? And then who knows?
Oh, wow. Well, that's fantastic. Great advice. And yeah, I feel the same. There's so much that we don't know, and so much to know. And there's never a boring day, because there's just so much out there that we can explore and discover. So,
and if you have a day that you know everything, you're not asking the right questions, right?
That's right. Well, Dr. Paig-Tran, thank you so much for being with us today, and again for your time. We are going to say goodbye to everyone, and thank you all for joining us today. We had a wonderful experience, and talk to you all soon. Thank you.
Bye. Thanks for having me.
Thank you for having it. Thank you. Bye.